hearing from Dr. Gidon Felsen, and Gidon has been a fun individual to work with in the time that I've been here. I'm sure he was fun before that too. But every time I have meetings with him, he's the PI who always comes up with the most challenging questions for administrators. Can my fellow compatriots attest to that? It is. You keep us on our toes. That is true. Indeed. So Gidon joined the faculty here in 2009, and in 2016 he was promoted to associate professor. And he got his PhD from the University of California, Berkeley, as well, which is supposed to be a pretty good school from what I hear. So, yeah. And Gidon does some really fascinating research, and even since the last time he did this presentation back in 2013, yeah, it was. He is kind of taken on some new directions too, but I always call it he's the PI most likely to take over the world, and you'll understand why here in a little bit as he gets into the gist of it. So, I'll turn it over to you.
That's a tough introduction to live on. Yeah, so I study how the brain makes decisions and then acts on those decisions. In the sort of the context of the department, we span a lot of neuroscience and some non-neuroscience physiology. I'm very much in the neuroscience camp, and I'm kind of at one end towards the systems and cognitive side of things. So this might be a little bit different than what you've heard about before. And to give you just an idea of the overall sort of question that I was interested in, as I said, it's how the nervous system makes and acts upon decisions. Because many of you are familiar with grants, I'll quote from the Brain Initiative to give you an idea of sort of the level at which I'm interested in addressing this kind of question, because there are many levels at which you can address an open-ended question like this. So I'm interested in understanding these neural circuits that are involved in these decisions and actions. And if we want to truly understand a circuit, that requires identifying and characterizing the component cells or neurons in that circuit, defining their synaptic connections with one another, observing their dynamic patterns of activity as the circuit functions in vivo during behavior, and perturbing these patterns to test their significance. So the things that I highlighted here, identifying and characterizing specific types of cells, defining their connections, recording their activity during behavior, and perturbing or altering that activity during behavior. Many of these things are what I do. Like, you could even say I do all of these things. There's a little bit less on the synaptic connections than all of the other aspects. So I kind of wanted to give you a flavor for the sorts of experiments that we do in the lab. Obviously, feel free to ask questions at any point. This can be formal. So to take a high-level view of what the components of decision-making are, I'll use an example that probably we're all familiar with. If you're in a cafeteria, you might be deciding whether to have pizza or salad. And so your decision is whether to reach for the pizza or the salad as you're going down the line and take it, right? So there's a decision, and that's based on various factors that we'll get to in a second. You're going to make some action, take the pizza or take the salad. And what are those factors? Well, you've got some behavioral goals, right? So maybe you want to be happy. Show that little happy face. Maybe you also want to be healthy, and effort to reach out to the more cardio-oriented people in the department who use a healthy heart. Okay, so those are the things, some of the things you're going to consider. Now, based on previous experience, you know that the pizza makes you happy, but it's less healthy for you. The salad, you're not as happy with in the immediate moment, but it's probably the healthy option in the long run. Right? So that's going to contribute to what decision you make. There's various other factors that are beyond the scope of this talk, let's just say, various other brain states that are involved. Of course, after you make your decision, there's some outcome, right? You take the pizza, okay, I'm happy. That wasn't so healthy for me, and that's actually what contributes to how you make these associations in the first place and will affect your decision the next time. Okay. So one thing I want you to notice is that it's not that hard to make sort of a little flow chart like this with these different functions in different boxes, right? You represent the sensory information. This is the kind of thing that Joel studies, how's visual input represented. There's some motor output, right? You have to move to grab the pizza. Roughly speaking, maybe that's what Abby studies. And in between those, what I'm calling the decision in this case is selecting the action, right? Which action do you want to do? Grab the pizza, grab the salad. So it's really easy to put these in different boxes, but the brain doesn't necessarily work in these sorts of boxes. In fact, there's a lot of overlap between the roles that even the same brain area will play in all of these different aspects. Okay. So how do we study decision-making in the lab? One important concept is that we're interested in spiking activity of neurons. So you've probably heard before that neurons fire action potentials, depolarizations that can travel long distances. This is sort of the currency of information and computation and transmission of information in the nervous system. So as opposed to many of the labs in this department, I'm not so interested in what's going on inside the neuron. I'm interested in whether it's firing a spike or not, and when that spike happens and how many spikes there are. So I kind of abstract a lot of, like, you may have seen some interesting electrophysiology traces that are the difference in voltage potential between the inside and the outside of the cell. All I care about is when that voltage potential is so much that it causes this sort of big spike. That's what actually gets transmitted from one area of the brain to the other. All right. So in order to study how sensory input is transformed into motor output via decisions, we have to know enough about how sensory inputs are represented and how motor outputs are represented. So to study sensory inputs, classically what experimenters have done is shown a lot of different types of stimuli. In this case, these are just gratings at different orientations. And you can record from a single neuron and look at how it responds to those gratings. And you'll find, this was Nobel Prize winning work in the late 50s and early 60s, that neurons in the primary visual cortex back here are tuned for a certain orientation. So they'll fire a lot of spikes, let's say for this horizontal orientation, and many fewer for a vertical orientation. Right? So that was Nobel Prize winning because it was the first clue that there are individual neurons that are really representing the world out there. And you can take kind of the flip side and look at how spiking activity in some other region of the brain corresponds to movements. And a lot of the movements that have been studied are simple movements like mind movements. Okay? And again, you can say that a neuron will fire a spike or multiple spikes for movement in a particular direction and fewer spikes for movement in another direction, let's say. Okay. And now that the whole field has some idea of how sensory input and motor output are represented, we can start to get at how you go from one to the other via decisions. Okay, so that's like the really, really big overview of how this field works. And the way that we kind of approach this in my lab is that we record and manipulate activity in specific regions and even in specific types of neurons in mice while they're performing a behavioral task that requires them to make a decision and then implement a movement based on that decision in order to tell us what decision we made. Okay, so why do I use mice? These are really valuable animal models in general, but especially for decision making, so they can learn decision making tasks really quickly. We can do recordings from them. We can manipulate activity in their brains. So this should be reminding you of the brain initiative quote I put up in the first place. Behavior is important, recording activity is important, manipulating activity is important, and also they're relatively inexpensive. A lot of the work that all this is based on is in primates, but it turns out that mice can do similar tasks to primates as long as you design them with what the mouse likes to do in mind. And we can actually go further by using some genetic tricks that are much further developed in mice than they are in primates. Okay, so like I said, I use behavior in my lab. I use a few different behaviors, but the one that I've used the most is an olfactory discrimination task, and unfortunately the video is not going to. So, and I'm not going to act it out. I'm just going to have to describe it. So here's a mouse. Okay, it's in a little box with 3 ports. It's actually in the center port right now. And what it does is it pokes into that center port, and gets a mixture of two different smells, call them apple and banana. And if there's more apple in the mixture, it learns to go to the left water port to get a water reward. And if there's more banana in that mixture, it learns to go to the right to get a water reward. So the decision that it's making is, should I go left or should I go right? And that's largely based on whether there's more apple or banana. In fact, it should entirely be based on whether there's more apple or banana. It turns out it's also based on some other things like what choice it's made in the recent past and whether it got rewarded or not. Okay. So the question that it's trying to ask is, should I go left or should I go right to get water? And it likes to do this because we don't give it as much water outside of the task as it would like to get. So it's motivated to do the task. All right, so just another little view of that. Or odor A goes to the left, or odor B goes to the right. And we have, a nice thing about this task is that we can time all of the various events. So we know when the mouse enters the odor port, we know, and then we can turn the odor valve on to release the odor at a certain time. We then actually make the mouse wait for this kind of gray period here until a go signal, and that's just an auditory tone. And then when it here's the go signal, it's allowed to leave the odor port and go into the water port, get a water reward. And the reason that we have this go signal that makes them wait, that we want to separate the time that they're making the decision of whether they're going to go left or right from the time that they actually initiate the movement. Because as I mentioned before, a lot of these brain areas are involved in both of these processes, so we want to kind of tease those apart. Okay. So mice can do this task really well. I'm just showing you a couple of examples. This is a wild-type mouse, and this is a transgenic mouse that's expressing this Cree protein in a certain type of neuron. We'll get to that a little bit later. So what you're looking at are called psychometric functions, and you're looking at the fraction of trials on which the mouse chose the left port as a function of the percentage of odor A in the mixture. So when there was all odor, 100% odor A, the mouse was supposed to go to the left, and it does almost all the time, right? Fraction of one. When there's 0% odor A, meaning 100% odor B, they always go to the right. And in between, they sort of distribute their choices, okay? And so these sorts of psychometric functions are going to be really useful for us, not just in showing that they can do the task, but in when we start manipulating activity and seeing how it affects behavior in a couple of slides. So decision-making is happening in a lot of different parts of the brain. So this is the slide that's supposed to overwhelm everybody to show that there's no way that we can possibly study this kind of process. So all of these different boxes are brain areas that all have some known role in this case, visual decision-making. That's what's been studied the most. So how do we decide where we want to focus, given that current technology, even though Joel Zilberberg would really like this, we cannot record from every neuron in every brain area at the same time. We have to focus somewhere. And so I've been focusing on this area here, abbreviated SC, which is called the superior colliculus. Okay, so it's relatively close to the motor output, which in this case is a saccadic eye movement. That's just a directional eye movement. So it's only a couple of steps away from that movement. It's not like in the cortex, which is what we like to think of as the area that makes us smart and is doing a lot of stuff that we do, and especially with respect to decision making. But it's known that this area, the superior colliculus, is involved in producing motor outputs, and there's increasing evidence from my lab and others that it's also involved in selecting which movement to make, which is how I'm operationally defining the decision in this case. Should I go left, should I go right? Okay. And taking a step back here, I start by talking about decision making, and we make decisions every day. It's a very high level process. And now I've got this mouse doing what looks like a really simple task, right? That's because mice can't they can't make the kinds of decisions that we might be really interested in, right? What college should I go to? Who should I marry? Things like that, right? Mice are not, that's not what they do. We also want to have something nice and well controlled, right? So the decision that they're making here is left or right. Okay, so this is the way that we're studying it in the lab. So to give you just another view of the colliculus, so here's a parasagittal section of the brain helpfully labeled, front, back, top, bottom. The colliculus is over here. Okay, so like Abby studies this area over here, the cerebellum. Nathan and Suk study factory bulb. Joel's focused on the visual cortex over here. Dan's in the inferior colliculus, obviously worse than the superior colliculus here. So just to give you some idea of like where we're all focusing on in the brain here. And so what do we want to, how do we want to determine the role of this particular area or any area in something like decision making, right? How do we actually study that relationship? There's three things that we try to do, and this will also kind of refer back to that initial slide. So the first thing we want to do is record activity during behavior. We want to see whether activity correlates with a particular decision, right? Like choosing left or choosing right in this task. The next thing we might want to do is elicit activity, right? So create activity that wasn't there before and see if that changes the decision, right? Finally, we might want to silence activity and then see, again, if that changes the decision. If you silence activity and that decision doesn't happen anymore, you can say that that activity, and therefore that brain region, is necessary for the decision. Okay. So we want to record it and you want to manipulate it. And it's useful to manipulate it up, increase it activity, or manipulate it down, decrease activity. So how do we do that? Right? And this is, so again, this is sort of the bread and butter of what my lab does. Right? We do it in a few different contexts. I'm just going to give you a flavor for some of the experiments, particular experiments that we do. So how do we record activity? We do that with an implanted drive that has tetrodes, which are 4 wires that are bundled together. So here's what a tetrode looks like. If we zoom in on the end and turn it around, you can actually see that there are 4 different little wires in that. So these are lowered into a particular brain area with these screws that you can kind of see up here. The eagle-eyed observer will notice that this is not a mouse, this is a rat. It just happens to be a better picture that I had. This is recordings that I did as a postdoctoral fellow, actually. And so we can basically pick up the spiking activity of not just one neuron, but little groups of neurons on the same tetrode, using the fact that we have 4 different leads. So basically, we're getting slightly different signals, different voltage signals from different neurons that happen to be near these leads, and they're going to give slightly different profiles of voltage change on the different leads, and that allows us to kind of tease apart which neuron is firing which spike. Okay, you can kind of think of it as triangulated. You've got some source, and you've got, in this case, we're quadrangulating, we've got 4 leads. So there could be a few neurons that are nearby, and we're going to record from all of them at the same time. And then we've got a few neurons in there. A few tetrodes, all targeting the same area. And the kind of data that we get, so we'll skip a lot of the pre-processing, but the sort of data that we get and how we combine it with the behavior looks like this. So these are called raster plots. Each line, each row of the plot is one trial in the task. And the little black tick marks are the spikes that a single neuron, one individual neuron, fires. So again, we might record from a bunch of neurons, but we're going to look for this first pass, we're going to look at just one neuron at a time. Okay. So each row is a trial, the black tick marks are spikes. I've aligned all the trials to a particular time in the task, and that's the time that the odor valve opens. That's when it delivers the odor to the mouse, the A and B odor, and it's when they can start to make their decision of what odor is this, and therefore should I go left or should I go right? Okay, so that's the orange line here. The green tick marks show in each trial when the go signal happened, and then the blue tick marks show when the mouse left the odor port. All right, so in between the orange and the green is when they're making their decision, and then they start moving when they leave the odor port. All right, now I'm also grouping these trials by what choice the animal, the mouse made. So for all of these trials, the mouse made what I'm calling ipsilateral choice. I'm only recording from one side of the brain. It happens to be the left side of the brain. So the ipsilateral side, the ipsilateral choice is the choice on the same side as where I'm recording. So that's a left choice. And so you can see that there's some activity on those left choices after the odor valve is opened. But you can see that there's a lot more activity at the exact same time in the trial, but when a contralateral or rightward choice is going to be made. Right? So this is left, and this is right. You can disregard the fact that we have two different delays here. That was, again, to kind of separate out the time of the decision from the time of the movement. Not super important for this purpose. But the point is that there's a lot more spikes preceding a rightward movement than preceding a leftward. You can see it even better in what's called this peristimulus time histogram. So in red are the trials where the mouse chooses the right port, and in black are trials where the mouse chooses the left port. So there's a lot more activity, right, firing rate over time. So a lot higher firing rate as the mouse is making that decision when it's going to be moving to the right than to the left. Okay. So if I were recording from this neuron and watching the mouse, I could actually predict which way it's going to go based on the activity of this particular neuron. If it fires a lot, right, then I know that it's going to be going to the right, and if it doesn't fire very much, it's going to be going to the left, right? Because keep in mind that it hasn't moved until the blue tick mark, right? It's just in the center port. Okay, so this gives you an idea of how we can show that activity of neurons is correlated with a particular choice, in this case, a rightward choice, right? And notice that it's not the case that it's simply firing to make the mouse move to the right, right? Because it's actually, its activity decreases right when the mouse leaves the odor port. It's really firing building up to that point of leaving. So it's corresponding to the time of the decision and not the time of movement. Okay. So that's a correlation, but in science we don't like correlations, we like causation. We like to know what caused something. So that's where manipulating activity comes in. And so there's a few different ways to manipulate neural activity. The way that we and many, many other labs have started using in the last 6, 7, 8 years is called optogenetics. I'm feeling that this is what Ron was talking about when he said that my lab is going to take over the world. So this is a cool technique. I did not develop this technique. I just happily use it. The idea is that there are proteins that when they're hit with light of a certain wavelength, they can be expressed in neurons, and they are actually a channel that allows cations, positive ions, to flow into the neuron. And that causes the neuron to depolarize and eventually fire an action potential. So if we can get this protein, which is called channel rhodopsin, into a neuron and then deliver blue light, it will cause the neuron to spike. And these proteins are found in nature in various algae and other types of organisms that what they do is they navigate towards light at a certain wavelength to find their food. And using tricks of molecular biology that, again, I did not develop, but I'm happy to use, we can express those in certain neurons and cause them to fire spikes. So this is just an early example showing that this works. When you deliver blue light, which is these blue pulses, and you record from the neuron, you see reliably this spiking activity of the neuron every time you deliver light. So what we can do is express that protein in neurons in the superior colliculus and in some particular region of the colliculus, which is what I'm showing here. And it's labeled with a fluorescent protein so we can see it in green in this image. And then we actually implant an optical fiber, right, that's targeting that area where the neurons are expressing this optogenetic protein. And we can deliver light through this fiber either via laser or via LED, and it will cause neurons to fire just like this. Okay, so this is just sort of a schematic of, okay, you've got your light source, you're delivering light through your optical fiber, delivering light directly into the brain and causing activity in these neurons. This is a really cool technique, and this is also the basis for at least the origin of the optogenetics and neural engineering core that Abby and I directed, and you've probably heard about. We try to help people incorporate this sort of technology into their experiments. Okay, so we can use this to manipulate activity, either increase it or decrease it, and see if that changes behavior. So if we inhibit activity in the left colliculus, we're doing that with a different optogenetic protein that instead of making the cell fire, it actually makes the cell fire fewer action potentials and silences the neuron. And we can see that we shift this psychometric function between trials where we're activating, well, inactivating those neurons with light, trials where we're not. So on half of the trials, we deliver light. That's the solid line. And on the other half of the trials, we do not deliver light to these neurons that are expressing this inhibitory opsin. And what you can see is that we're delivering it to the left side. And so we're causing the animal to make more leftward choices with the same percent of the left odor, right? Remember, they're still doing this odor A, odor B task, but we're biasing them to go to the left more often when we silence the activity of these neurons. And that makes sense because I showed you an example neuron a couple slides ago that was that had more activity when the animal made a rightward choice. What I didn't show you is that we've recorded from many, many more than just one neuron, and that's generally true, that these neurons basically signal a rightward choice. Of course, there are neurons in the right colliculus, because there's two colliculi in the brain. Neurons in the right colliculus that are doing the opposite, they're signaling a leftward choice. Okay. So we showed that activity was correlated with rightward choices, and now we show that when we turn off that activity, we have fewer rightward choices and more leftward choices. So that's some evidence that there's a causal relationship between the activity and the decision. Rob, you don't look totally convinced. You kind of were like, I don't know, maybe.
No, I was just like, shake your head like, wow.
All right, so then we can do.
Doubting your research.
You wouldn't be the first. So then we can do the complementary experiment. We can excite neurons in the left colliculus using another optogenetic protein, the first one that I showed you, channel rhodopsin 2. And so here, light off is in gray and light on is in black. And you see that we can shift to that psychometric function to the right. So when we increase the activity of these neurons, we're making the mouse choose the right side more often. Right? So sort of necessity, sufficiency arguments. Also interesting that we're not just affecting all of the choices equally. So we can separate out the trials into the ones that are relatively easy for the mouse, like when it's all of the right odor, or B, or all of the left odor, right? They almost always get those correct, right? They go left all the time or right all the time. But these trials in the middle are more difficult, right? More difficult choice of whether they should go left or right. And it turns out that those are actually affected more by the light when there's a small odor contrast, a small difference between odor A and odor B. Those are affected more than the easy trials. You can kind of see that in the psychometric function. Even when the light is on, they still get these easy trials almost entirely correct. This example. So this is more evidence that this particular area, the colliculus, is not just involved in initiating movements, which has been known for a while. It's actually involved in selecting these movements and making the decision to move left or to move right. Okay. So that's some sort of high-level evidence that the colliculus is critical for these sorts of decisions. And one of the directions that my lab is going now is try to figure out the mechanisms by which it's doing that. So how is the colliculus actually contributing to this decision, to the selection of action? And there's a couple of things that we've been thinking about, and there are various models that have been proposed, not by me, but by other people, that involve competition between the two colliculi. So this is just a schematic of the left colliculus and the right colliculus. And there's this idea that they're actually competing with each other, where the left colliculus is saying, go right, and the right colliculus is saying, go left. And the winner of that competition determines the decision that's made and the movement that's ultimately made. Okay. And so there's this idea that inhibitory neurons, that's what the I stands for here, mediate this competition between the left and the right colliculus. There are also ideas that there's competition within each of the two colliculi. What I didn't explain is that in different parts of the colliculus, neurons have a preference for movements to particular regions of space. And in fact, in the more rostral forward part of the colliculus, neurons prefer very small movements, even going to no movement at all, just fixating. And in the more caudal part of the colliculus, back here, they prefer these saccadic movements, these large movements. And again, you see that there's perhaps in the schematic some competition between them, mediated by these other mysterious neurons that are proposed to be inhibitory neurons. Okay. So This is the kind of stuff that people have been proposing and thinking about for a while, not just in the colliculus, but many, many different brain areas. It's kind of a high-level question of how do areas interact and how do they make decisions between this idea of sort of competition between brain areas is very prominent. Colliculus is just a convenient, good place to look at it. So we're interested in how this might happen in the colliculus using some of the tools that we have. Now, another thing to note about the colliculus is that it has a lot of different types of neurons that are all kind of right next to each other. So this was an image from Ramon y Cajal, one of the top 2 neuroanatomists of all time. This is from like the early 1900s. Just sort of showing a diagram, a sketch of all the different shapes of neurons that are involved. We now know a lot more about these neurons. We know that they're excitatory neurons, they're inhibitory neurons, they're neurons that project to different brain areas, and all of them are kind of jumbled together, right? And that means that the methods that we use, like when we drop our tetrodes or electrodes into the brain to record from them, we actually don't know what type of neuron we're recording. And with most of the classical techniques, we don't know what type of neuron we're manipulating when we manipulate activity. Okay. This is true in just about every single brain area. The brain is just really complicated, right? Different types of neurons going every which way, releasing different types of neurotransmitters. So we need some kind of cell type specific methods to start to address what these specific types of neurons are doing and how they might be contributing to these sorts of models. Okay? And that's exactly where optogenetics comes in, right? So I already introduced what this idea is. We're starting to use it now to record from specific identified types of neurons. Instead of just taking whatever neuron happens to be near our electrodes or tetrodes, we're going to target our recordings to specific types of neurons. And we do that by expressing that optogenetic protein, channel rhodopsin, in one particular class of neurons. In this case, what we're starting to do first is express them in inhibitory superior colliculus neurons. So I'm just kind of sort of showing the colliculus as the circle and the different types of neurons with these different shapes, excitatory and inhibitory. And I'm showing these inhibitory neurons in yellow because they're expressing the optogenetic protein. So then what we do is we have our tetrode drive, right, that we've been using to record from neurons, and many people have been using to record for a while. And now we're incorporating this optical fiber with light source. And when we deliver light to the neurons in the vicinity, we can see which ones respond to light. Okay, so this should look like a slide from an image from a couple of slides ago. When we deliver these pulses of blue light, some neurons we see regularly fire spikes in response to those pulses of blue light. And those are the ones that we now know are expressing channel rhodopsin. Because we only put channel rhodopsin in the inhibitory neurons, we know that that's an inhibitory neuron. Other neurons, in fact, most of the neurons we find do not respond to the light. And so they're either excitatory neurons, or they might still be inhibitory neurons, but they're just not expressing the opsin. So we can't say what neurons those are. We're in the same boat with those neurons as we've been for many years. They're an identified neuron type. But the point is that with some of these neurons, we can actually know what type of neuron they are. And so a few labs are starting to use this sort of technology, as are we, and in particular, Jackie Essig in my lab has started recording from identified inhibitory neurons. So I'm skipping a lot of the details, and I'm just showing you one example. The point is that we can get the same kind of information, the same sorts of rasters, PSTHs. But now the key piece of information that we have is that this is an inhibitory neuron, not just any old neuron. Okay? So she's first targeting inhibitory neurons, but she's actually in parallel has started to do some experiments where she's expressing channel rhodopsin in neurons in the colliculus that are known to project to a certain brain area and are thought to have a certain function, but nobody's ever studied them because how do we identify them in a behaving animal? So now we're starting to be able to identify those and again, record for them during these kinds of simple decision-making tasks. Okay. So we're basically hoping to answer some of these questions about how actions are selected in this area and use that really as a model to understand how more complicated decisions are made in even other brain areas. So that's kind of one direction. Now, I showed you the overwhelming slide before that had many, many different brain areas on it. And the colliculus is just one of them. So I actually am interested in many of those other brain areas. So my lab sort of started by focusing on the colliculus, but we've also done a lot of experiments in a region of the basal ganglia called the substantia nigra pars reticulata that inhibits the colliculus. Right? Mario Lintz did a lot of this work. Beth Stubblefield is working on this in a collaborative project at Geneli Research Campus. That this is one of those fun questions, I guess, that Ron mentioned. We talk about this collaboration quite a bit. Beth and also former postdoc John Thompson, still a collaborator, has been looking at another area called the pedunculopontine tegmental nucleus. This provides a cholinergic input to the colliculus. Okay. Jackie Essig and I wrote a short review paper on a dopaminergic input to the colliculus. And I've started working with Abby Pearson and Joel Zilberberg on cerebellar input to the colliculus and how that's contributing to the decisions that are made here. So the point is that I'd like to, we have some idea of what the colliculus is doing. And now I'd like to drill deeper down with the colliculus by understanding the mechanisms by which specific cell types are performing this decision-making function. And in parallel, we're trying to figure out how all these other areas are modulating collicular input, modulating the computations that are being performed. I should also say that also in collaboration with John Thompson, who's now in neurosurgery, we're doing, roughly speaking, some similar experiments in people. Right? So John has access to patients that are undergoing deep brain stimulation for Parkinson's disease, and they have to, as part of that, there's a surgery where they get an electrode implanted into the brain. And the idea is that when you deliver current through that electrode, you can actually relieve some of the symptoms of Parkinson's disease. As part of that process, you've got a patient with an electrode in their brain so you can record activity. And many patients, very surprisingly, are willing to do simple decision-making tasks. Much like what we have our mice doing, we actually have an auditory task that's just practically a lot easier in the operating room. But they're making a decision about the pitch of a tone and moving a little joystick to report their decision. And we're recording, in fact, from this substantia nigra pars reticulata area in humans. So that's really exciting. Something that I did not think I'd be doing until I got here and realized that that's what happens in the hospital. We're doing that with an MD PhD student, Andy Teckerwall, that's working in both of our labs. So yeah, hopefully that gives you a flavor for some of the research that I do. I also just wanted to mention, I do something else that's totally different, and Rob kind of alluded to this as well. So this, I don't think anybody else in the department is thinking about public policy, but I am. So I'm interested in how neuroscience can inform public policy, and specifically policies that are designed to help people make better decisions. So it's probably no secret that we humans make predictably poor decisions, right? Like we will reach for the pizza even when we should reach for the salad, right? This is maybe exemplified best in this popular book called Nudge by Cass Sunstein and Richard Thaler. So Richard Thaler just won the Nobel Prize a few months ago for his work leading up to this book. And the idea is that if you set up the environment in such a way to encourage or promote particular decisions to be made, you can get those decisions to be made more frequently. And that will result in, as they say in the title of the book here, more health, wealth, and happiness. So one classic example is we humans have a tendency to go with a default option when there's multiple choices. If there's a default and we're not really experts on this thing, we're like, yeah, the default is good, right? This is what you do when you update your computer, for example. You probably don't override the default. And so there's this idea that may be familiar to some of you as administrators that if you default people, workers, employees into retirement savings, into putting money into a retirement savings account, they're more likely to put money into that retirement savings account, right? And that's probably a good thing because most people don't save enough for retirement, right? Now, an important feature of this is that you're not, in this default case, you're not saying that the person can't make the other decision, right? The default option might be put a certain amount in retirement. They could still override that default and say, no, I don't want to put anything in retirement. Okay, so you're not actually limiting the choices they can make, right? It's not like a heavy-handed, like you have to do this. Now here at our university, I think it is, you have to do this. Let's leave that aside for a second. So this is an example of how to set up what's called the choice architecture to promote making one certain decision without taking away any choices, right? So, I read this book, it came out about 10 years ago, and I said, so they're talking about poor decision-making and how we can kind of fix that and how we can counteract that. And it's largely based on sort of the psychology of decision-making. And I study the neuroscience of decision-making. So I wonder whether neuroscience can contribute to this sort of a thing. So that's what I've been spending some time thinking about. And this is just sort of a schematic from a recent paper that a former grad student and I wrote that describes how we make poor decisions, and that leads to things like poor health. If we introduce some kind of decisional enhancement strategy, like a nudge, that will lead to improved health. There are several ways that neuroscience can contribute to those sorts of decisional enhancement strategies. First of all, as I mentioned, most of these are based on the psychology of decision-making, what you might call behavioral economics. This is what Richard Thaler actually won the Nobel Prize for. So basically, this is the point that humans are they don't make the optimal decision that an economist would predict, that traditional economists would predict a person would make. They have various cognitive biases, right? So they go for the default option, for example. Or another good example is that people are biased towards sooner rewards, even if they're smaller, rather than larger rewards later on. Again, think of, you want the pizza right now, even though the salad is going to be better for you in the long run, right? So these cognitive biases, neuroscience can actually inform sort of the mechanisms for how those biases happen. Okay? A lot of the criticisms of these sorts of nudge type strategies is that they violate people's autonomy. You're forcing me to choose something. Even though, as I mentioned, you're not actually limiting the choices, but people feel manipulated, right? When, especially if you find out that that's how things were set up, or, to go back to the cafeteria example, what a lot of stores and cafeterias do is they put the healthy food closer than the less healthy food, right? So you're being subtly manipulated, maybe for your own good, but people are not super comfortable. So I've actually done some work looking at how neuroscience can inform our concept of autonomy, which then in turn, all of these things inform a set of potential strategies for making better decisions. And then this is something that I don't do at all, but other people do. There's a whole field of this. Once you have a set of potential strategies for helping people make better decisions, you can actually use fMRI, functional imaging, to look at how brain activity correlates with the decisions that are made under those sorts of strategies. And that can help you narrow down which is the most effective decisional enhancement strategy. So just one other example of how I'm trying to link these two things, that again takes us back to our initial example with the pizza and the salad. Now I'm going to have to apologize because in this paper we changed it to brownie instead of pizza. But same idea. You want the brownie now, right? It's got sugar, even though it may not be the most healthy thing. So there's a very prominent idea in neuroscience of decision making that there is a decision variable that's represented in the activity of a neuron or a group of neurons. It could be anything. It's a little bit abstract. But that decision variable actually is what determines what choice you make. And that decision variable changes over time. Okay, so it kind of bops around, right? It's a little bit noisy here. It's bopping around, it's going down, it's going up, right, in time. And what do down and up mean? They just mean more evidence for choosing one thing, let's say the salad, over choosing the other thing, the brownie, all right? And the idea is that once this decision variable reaches one of the bounds, that's when the choice is made, and that is the choice that is made. So in this case, the decision variable is bopping around. Why is it changing in time? Maybe like you're starting to smell the brownie more at this point in time, right? Maybe like as you're talking to your friend, you're not really thinking about the decision, but then you actually put some time into thinking about it. So you can kind of think of this, again, as a competition, right? Choose the brownie, choose the salad, right?
there's a lot of factors in play, and this decision variable summarizes what choice you're going to make. So you haven't made a choice until you reach the bound, and then you make a choice. Okay. So in this case, look, you chose the brownie because you reached the brownie bound. So how would a decisional enhancement strategy like nudging, how can we think about that in terms of this sort of a model, which is, again, hugely prominent in neuroscience? Well, one way you can think of it is that it's actually changing your starting point. Okay, so we've moved your starting point closer to the salad bound. And now if you have the same pattern of activity, I just took the same trace and I shifted it up a little bit, it would actually reach the salad bound before it could then go back down and reach the brownie bound, right? So you've made it more likely that you're going to have the salad, right? You could also equivalently think of it as keeping the starting point in the same place, but moving the salad bound closer. Decision, same thing. Okay. Have you precluded the possibility of choosing the brownie? Not necessarily, right? Using top-down influence in your brain, your brain can move those bounds, right? It's a little bit hard to see, I guess. But you can actually shift those bounds further away, which basically means I want to give it a little bit more time before I make the decision, right? And this might be, you saying like, hey, I don't want to be, I realize that I should choose the salad, and I usually choose the salad, but I'm going to have salad tonight, so it's really going to be okay if I have the brownie, right, or pizza. So by moving these bounds, even though a nudge has occurred, you can still be free to choose the brownie if you would want to, right? Again, captured by this kind of a model. Another way you can think about it is that nudges can kind of debias the biases that we naturally have. So like I said before, we're biased towards the immediate reward now, even if it's less good in the long run than some other things. We want the brownie, we don't want the salad. So the way you can think about it is that maybe we're already biased by our very biology to choosing the brownie. So a nudge is kind of counteracting that bias and making it easier for us to make the better choice, choosing the salad. So that's just a flavor of this other side branch of research that I've been working on. So, of course, I want to thank everybody in the lab, past and present, who's been working on all this stuff. You in the department know all these people. I also want to thank the department itself. It's really been an amazing place to work, especially the awesome administrative staff. We researchers couldn't do what we do without support that we get from you guys all the time. So we really appreciate it. And yeah, let me know if you have any questions.
I have a question. So you're competing renewal on your R01. Is that focusing in on the particular neurons that you were showing us earlier and
yeah,
zeroing in on that piece of it?
Yeah, it's exactly this point. It's looking at excitatory and inhibitory neurons in the superior colliculus separately to try to figure out like how to set up these sorts of models to understand how the colliculus does what it does. That's its direct thing. Yeah.
Did you talk about your collaboration with the Center for Ethics and Health?
Yeah, so that's this last stuff. So yeah, right. So I have funding to do this for the next few months at least from the Greenwall Foundation. I love their initials, by the way. And this is a bioethics, well, mostly a bioethics foundation. So how does this bioethics work? I think it fits into bioethics because basically the poor decision making that we are known for leads to poor health. Okay. That's one way. It's a bioethics issue. The other way is that I really have been interested in this relationship between neuroscience and autonomy. And autonomy is kind of a core concept in bioethics. I'm actually interested in more of its sort of role in fundamental philosophy. Right? People have been thinking about what autonomy is and what it means for literally millennia, but it really comes down to like the decisions that we make, right? And that's something that we're learning a lot more about with modern neuroscience. So I'm trying to apply what we know about neuroscience to autonomy. And then I think, as I tried to show here, that also, right, that also relates to this, these questions about nudges and other decisional enhancement strategies.
So is a nudge like just education? You're educated on what the choice
Yeah,
would
right.
be?
Right, good question. So it's sort of difficult to pin down exactly what a nudge is, but it's generally agreed that it's something that you sort of that operates covertly. It's the way people usually think about it. So you're not necessarily aware that the brownies are further away from you than the salad in the cafeteria. And if you were aware of it, now I'd still consider that some other decisional enhancement strategy, and education would be a decisional enhancement strategy, but it might not be a nudge, strictly speaking.
So it seems like there's a lot of overlap with modern marketing, right? In terms of
Oh yeah,
everything from product placement to
right. It's almost
social media to where
advertisers-
when you go to the grocery store, what's at eye level? When you're picking out, whatever.
That's exactly right. So advertisers have sort of known about this and been exploiting it for a long time, right? And then it's been sort of the public health community that said, well, we can use that too, but for good instead of for profit.
Department stores are all set up the same way based on how we can shop. So that doesn't matter what store you're going into, the departments that we go to most often are going to be to our left every time. Kind of fun.
And is there some known bias that women shoppers like to go to the left?
I think they just studied, like watching women walk into stores in what direction.
Okay.
Like they decided to go.
I go to the left.
And so they set up. Me too.
Yeah.
So they set up like.
We should look at your follicles.
Shoes and handouts and everything will always be to your left.
That's my wife.
Mhm. So if you try to go to the right, it feels really weird. I've done that.
Very interesting. I
But
might listen to that.
do men go the other way?
Oh, I don't know.
We don't know where we go either. Let's go.
So what are you gonna do with this overall, once you get all this information? What is the big picture plan?
Oh yeah, that's a really good question. So on the basic research side, I unabashedly do basic research. I'm not trying to cure a disease in the immediate future. But my attitude, my approach is that we can't fully repair or prevent diseases until we understand how the system is working in the normal case first. And we really don't understand how the brain works, period. And that's kind of how I got into all this in the first place. And focusing on decision making is kind of the happy medium that I found, other people have found too, between this really high level question, how does the brain work, and something that we can actually study in the lab. So I'd say that there's a lot of research that has to be done at the level of individual neural circuits and types of neurons in those circuits, going again back to the Brain Initiative quote I gave you in the first place. And then there's a lot of theoretical and modeling work that has to put all of that data together. That's why we hired Joel, basically, right? This is a huge growing field of neuroscience, computational theoretical neuroscience, that takes a lot of the data that's being collected and puts it together into a comprehensive, integrated sort of model. And then, this is the long view, and then we can start talking about what's changing in, let's say, a disorder like Parkinson's and how we can repair it.
So do you, or anyone you know, do research, I guess, based on decision making when you're in a group of people? So instead of having the mice just by itself making a decision, if there's,
Peer pressure.
Social
what they make.
Yes. So yeah, there's a field of social decision making, and there's some work in mouse models on that. It's going to be you can kind of have mice play versions of games that experimenters have had humans play for a while, like game theory type things. Prisoner's Dilemma, for example, where I'm going to butcher this a little bit, but in the Prisoner's Dilemma, you've got two prisoners, and you can offer them, they can either rat each other out or stay silent. And if they both stay silent, then that's the best outcome for both of them. But if one rats the other guy out and the other guy stays silent, it's really good for the guy who did the ratting. So there has to be this cooperation that happens, right? So people have developed interesting analogs of that for mice. And then you can look at activity in different brain regions. Usually people are looking in, more of the cortical regions, and especially in like the regions that are related to emotion, like the orbitofrontal cortex. That seems to play a role in like individual differences even across animals of how cooperative they are versus not, for example. Yeah. So it's an interesting field.
Have you written anything yet into the area? You mentioned it's an area of personal interest as far as the overlap of autonomy, neuroscience, philosophy. I guess you could probably go so far as psychology and theology in terms of what does it mean to be human and freedom of will. Have you gone into that area yet in terms of your writing, or?
Only tangentially. So you could think of autonomy as kind of a possibly more tractable way of talking about free will. Because it's a well-developed philosophical theory. It kind of assumes that we have free will, and then it gets to the, what I'm interested in. But many people are interested in the question of how that could work in a deterministic universe. So I've avoided free will too directly. Like there was one exception that I wrote like a commentary paper on an article that somebody wrote that basically said that if everybody accepted the fact, assuming it's a fact, that we don't have free will, that society would like break down. So it's a useful construct useful lie that we tell ourselves. So a collaborator and I thought that was interesting, and we wrote a commentary about it and how, it's that might be the first order thing that you might think would happen, but there's so many other unintended consequences of finding out something so huge like that maybe would change so much else that it's hard to predict what the result would be. I got into all of this through the field of neuroethics, which roughly speaking is the ethics of neuroscience and also the neuroscience of ethics. The ethics of neuroscience is not so different than traditional bioethics, like animal research and like authorship on papers and not lying about your data. Like those are classic issues in scientific research. Neuroscience of ethics is more like we've developed these systems of ethics based on our intuitions and philosophers' intuitions and how we think about certain problems. We know that intuitions are often wrong, right? Cognitive biases. They're wrong in predictable ways and for predictable reasons. The brain, our brains evolved not in us modern humans, but they've evolved over millions of years in animals that were that were living in a very different kind of world. They evolved to survive, right? There's a reason that we want the fatty sugar food right now rather than the healthy food, because in most of human history, we couldn't just go to the grocery store and buy it, right? So it was good to take advantage of that when we had it. So it's a little bit of a tangent, but the point is that the way we think about ethics and things like autonomy are based on how our brains have been wired up to think. And they evolved to be wired up that way in a very different context than they are now. So it's kind of interesting to think about what underlies these ethical beliefs.
Well, we should try to close here. Thank you, Gidon.
Thank you.
Very interesting. And see,